The next presentation is going to be a presentation covering Chapter 3 information, which will cover information in 2 labs from your lab manual. The 2 labs that are covered in your lab manual will be biochemistry, structure of molecules, as well as chemical composition. Chapter 3 talks about organic molecules. Organic molecules are going to be carbon-based molecules, often containing a combination of carbon and hydrogen. Some examples of organic molecules are going to be molecules that you have heard about before: carbohydrates, DNA, proteins, fats are examples of organic molecules. Organic molecules are very diverse, and many organic molecules are made of something called a carbon skeleton. If you think of what the skeleton does for the human body, it may help you to understand what a carbon skeleton does for an organic molecule. In humans, skeletons allow us to attach other organs on, such as tendons, ligaments, muscles. Basically, the skeleton provides the structural support to attach other tissues and organs onto. We see something similar in organic molecules. Organic molecules have different types of carbon skeletons, and we are going to list them here. We will also make some small diagrams of carbon skeletons. The first type of carbon skeleton that we're going to talk about is called carbon rings. Carbon rings are made of carbon atoms that are joined together. It is not like the ring on your finger. It is going to look like a hexagon or a pentagon in shape and may have other shapes as well. Let's draw an example of a carbon ring. My first example of a carbon ring is going to be a 5-sided carbon ring. I'm going to draw a diagram that looks like a pentagon, where there's a carbon atom at almost every point within the carbon ring. Since the carbon atoms are joined together, we call that a chemical ring. So it is not like the ring on your finger, but in chemistry, this is called a carbon ring. We can draw another example of a carbon ring. I'm going to draw a hexagon shape. At almost every point in this hexagon, there is a carbon atom. And as you can see, one carbon atom is bonded to another carbon atom, which is bonded to another carbon atom, which is bonded to another one. So we have an almost complete ring of carbon atoms. This is why they are called carbon rings. Some carbon rings are small and individual. Some carbon rings are fused together. I'm going to draw what looks like a pentagon attached or fused together to a hexagon. A little bit of an error there, sorry for that. But this is what we call a fused carbon ring. So we have small carbon rings, we have large carbon rings, and we also have fused carbon rings. Again, where there's a carbon at most points within the ring. The other type of carbon skeleton is going to be called a carbon chain. We have small carbon chains. Looks like we have 3 carbon atoms connected together to form a small carbon chain. We have long carbon chains. a bunch of carbon atoms bonded together to form a long chain of carbon atoms. And we also have branched carbon chains. You can see it is similar to a branch on the tree. It is branched. It looks like a more complicated type of carbon chain. These are the skeleton of the carbon-based molecules or the organic molecules. As you can already see, there is a whole bunch of different types of organic molecules. We can just look at the different shapes of the carbon skeletons and determine that there will be a large variety of organic carbon-based substances. Let's move on to the next page. We are on question number 3. We are going to talk about the functional groups, which are sometimes also called the chemical groups that may be added to a carbon skeleton. There are a lot of functional groups, but we are going to look at 3 functional groups. The first one we're going to talk about is called methyl. Let's first describe the molecular formula for methyl. It is going to be CH3. How many carbon atoms are going to be in a methyl group? Looks like 1. How many hydrogen atoms are going to be in a methyl group? It looks like 3. The structural formula provides us with a little bit more detail. The structural formula shows us where the bonds are between all of the atoms within the functional group. So as you can see, the structural formula still has 1 carbon atom and 3 hydrogen atoms. Therefore, the structural formula provides us a little bit more detail. Methyl groups tend to be what we call hydrophobic. Hydrophobic is a type of chemical or a type of substance that does not like water. Our second functional group is going to be called carboxyl. It is sometimes also called a carboxylic acid. The molecular formula looks a little strange. It is made of 1 carbon atom, 2 oxygen atoms, and 1 hydrogen atom. Let's draw the structural formula. Okay, as you can see, there is something called a double bond shown in the carboxyl functional group. So this right here looks like 2 lines. That is a double bond. And everything else that you see with a single line are going to be representing what we call single bonds. Recall from chapter 2 that a bond is a force of attraction that keeps 2 atoms within a molecule in close association with each other. Carboxyl groups tend to be hydrophilic and acidic. So now we see that methyl group is hydrophobic, whereas carboxyl group is hydrophilic. Basically, carboxyl groups really are attracted to water or like water. So that makes it a little bit different than a methyl group. Our third functional group that we want to know is going to be called an amino group. And the molecular formula is going to be NH2. So it's going to have 1 nitrogen and 2 hydrogen atoms. Let's go ahead and draw the structural formula. There we go, NH2. Looks like they're all single bonds in the amino group. Let's take our methyl group and attach it to a carbon skeleton on the last page that we worked on. So let's say we attach a methyl group at the end of our long chain, CH3. That is my methyl group. That means that at that portion of the chemical, the organic molecule is going to be hydrophobic, since that was one of the characteristics of the methyl group. Carbon skeletons can attach to all of these different types of functional groups, which means that it results in organic molecules that are hydrophobic, hydrophilic, acidic, basic, and have lots of different properties. This is going to increase the diversity of organic molecules. We are on question number 4. We need to introduce some more terms. This term is called monomer, and it has that term mono in it. Mono means one or individual. Let's make a definition for monomer. A monomer is going to be one subunit found in a macromolecule. Macromolecules are DNA, carbs, proteins. Those are some examples of macromolecules. I believe we mentioned it in chapter 2. Let's give an example using the term monomer. I don't want to write that sentence. We'll say proteins are made of amino acid monomers. The other term is called polymer. So poly means many rather than mono, which means one. Polymer is a macromolecule made of many monomer subunits joined together. Joined together. Examples of polymers are proteins, which are polymers of amino acids, carbs, DNA, and then we will write etc. because we will give more examples. Let's move on to question number 5. We need to talk about 4 major classes of organic biomolecules. Now we have seen that there is a very large diversity of organic molecules. We're going to call the organic molecules that make up living organisms biomolecules. And examples of biomolecules are proteins, carbs, DNA, etc. These 4 classes are going to be examples of what we call biomolecules. So not all organic molecules make up living organisms, but organisms are made of organic biomolecules. The first major group of organic biomolecules is going to be called carbohydrates. Carbohydrates are sugars and starches. Carbohydrates serve as a primary source of energy for cells. That is not the only function for carbohydrates, but that is a good starting point. The monomer subunits that make up carbohydrates are called monosaccharides. The second major group of organic biomolecules is called lipids, commonly called fats, oils, and waxes. Lipids provide us a concentrated source of energy, and one characteristic that all lipids have in common is that they are some degree of hydrophobic. We're going to talk about the monomers that make up one type of lipid, which is called a triglyceride. A triglyceride is built by connecting three fatty acids plus a chemical called a glycerol. Our next category of organic substance is going to be called proteins. Humans have over 23,000 different proteins that do various functions. The monomers that make up proteins are called amino acids. This means that if we want to build a protein, we would need amino acid subunits to build that protein. Just like if we want to make a lipid, which is called the triglyceride, we would need three fatty acids plus a glycerol subunit in order to produce a triglyceride. Our last category is called nucleic acids. And some examples of those are DNA and RNA. The monomer subunits needed to make a nucleic acid like DNA or RNA are going to be called nucleotides. So at this point, you need to know the monomer subunit that is needed to make a specific substance. For example, I could say, what are the monomers that are needed to make up a nucleic acid? Your answer would be nucleotides. Or I could be even more specific and I could say, what are the monomers that are needed to build DNA? And your answer would still be nucleotides. We will learn about the structure of nucleotides, amino acids, the triglycerides, and monosaccharides somewhat within this presentation. Nucleic acids we will study in a little bit more detail at the end of the semester. You will need to know the structures and this information in order to get through the biochemistry lab from your lab manual. We're going to move on to the next question. Looks like we are on question number 6. We need to talk about two types of chemical reactions that happen in cells. There are two types of reactions that are going on in cells at any given time. Either your cells are breaking down substances or your cells are building molecules. And usually they are doing it simultaneously or back to back. We are interested in talking about the types of reactions that are specifically needed to make organic molecules, such as the substances that we talked about in question 5, like carbs, proteins, and lipids. The first type of reaction that we're going to be talking about is the type of reaction that is going to allow us to build. This type of reaction is going to be called dehydration synthesis, which is also called a condensation reaction. Let's explain what we mean by a dehydration synthesis reaction. An example would be when two monomers react and bond together to form a larger, more complex molecule. To form a larger, more complex molecule. I am going to draw a diagram of a reaction, which will include the carbon rings that we spoke about in question number 2 on page, in question number 2. I'm going to draw the carbon ring that we showed as a hexagon. So here's one hexagon, and I'm going to put a plus sign because it's going to bump into and react with another hexagon. So the individual hexagons are going to be called monomers. Remember that the hexagons represent something that we call carbon rings. I'm going to draw an arrow that's going to indicate a reaction. In this case, the reaction is going to be called a dehydration synthesis reaction. I am now going to connect the two hexagon or the two carbon rings together to show that the individual monomers were bonded together to form a larger, more complex molecule, which we're going to call a polymer. This is often called a condensation reaction because a water molecule is also produced when the reaction occurs. The next type of reaction is going to be when your cells are breaking something down. And this is going to be called a hydrolysis or a hydrolytic reaction. This is breakdown. So my example is going to be when we take a large complex molecule like a polymer and break it down into its individual units. Polymers broken down into monomer subunits. An example of this is digestion, when you digest your food. I am basically going to draw the opposite reaction as compared to the previous type of reaction. These two hexagons, which are connected together. This is our polymer. I'm going to draw the arrow that indicates the hydrolytic reaction. It's going to result in individual monomers. Basically, you're breaking the very large, complicated molecule down into the individual subunits. This would make sense when you think about digestion. The goal of digestion is to break your foodstuffs down into the smallest parts possible so that your intestines can absorb it and pass it to your body for energy. The hydrolytic reaction is called hydrolysis because during the reaction, a water molecule is broken down. So hydro is referring to water and lysis is referring to splitting. So during the hydrolytic reaction, water molecule is needed and it is going to be split apart. You will see this in more detail when you conduct your lab. Let's move on to question 7. We're going to go ahead and talk about those different types of carbohydrates. Remember that we had 4 major classes of organic chemicals. The first major group was called carbs. The second group was called lipids. The third group was called proteins. And the last group was called nucleic acids. You will have to know the following information in order to complete the lab activity. We're going to explain carbohydrates and break it down based on their size, which is reflective of their structure. The first type of carbohydrate is going to be called a monosaccharide. And it's going to be made of one carbon ring. Let me draw some examples. The first example I'm going to draw is in the shape of a pentagon. Remember that a pentagon is 5-sided. So here is my pentagon. I drew a simple shape called a pentagon, and this represents a 5-sided sugar that we call a pentose sugar. This is one carbon ring because it's all by itself and it's not connected or bonded to another carbon ring. Some examples of pentose sugars are going to be fructose, ribose, and deoxyribose. Deoxyribose is found in DNA, ribose is found in RNA, and fructose is a sugar that you probably have consumed. Let's draw another example of a monosaccharide. I'm going to draw a hexagon shape. Since this sugar is a 6-sided shape, we're going to call this a hexose sugar. One example of a hexose sugar is going to be called glucose. You will see different types of hexose sugar in the biochemistry lab manual from your lab book, and you should read about it and make sure you understand what you are looking at. In the lab manual, they do draw these pictures, which looks like hexagon and pentagon shapes. And anytime you see those symbols moving forward in your textbook or in your lab book, you should realize that these very basic symbols, which is a hexagon and a pentagon, represent simple sugars that we call monosaccharides. The second major type of carbohydrate is going to be called a disaccharide. This is made of 2 carbon rings. If you think about it, that term di means 2. So let's take the pentagon that we just drew up there, we'll redraw it, and we will connect it to the hexagon that we drew in the previous problem. So the pentagon is going to represent fructose, and the hexagon is going to represent glucose. And when you connect them together, you make a substance called sucrose, which is table sugar. This is an example of a disaccharide, and your lab manual has other examples of disaccharides, so you should keep an eye out for them in the biochemistry lab. Our last example, okay, is going to be called a polysaccharide. This is going to be made of many, that term poly means many, made of many carbon rings. So let me draw a bunch of glucose connected together. And I am definitely not an artist, so I do my best to draw. Although I don't do the best job ever, and I don't expect you to either, etc., etc. We'll say there's hundreds or thousands of these glucose units connected together to make a very large polysaccharide, usually hundreds or thousands. So an example of a polysaccharide is starch. This is made by plants and can be broken down for energy. Another example is cellulose, which is also made by plants, but serves as structural support and found in the cell walls of plant organisms. Our last type of polysaccharide let's go like this so we can read it, is going to be called glycogen. This is made by animals like you and I and basically provides energy to cells. So let's make sure we are all comfortable with the different symbols of monosaccharides or individual carbon rings, whether it's going to be a pentose sugar or a hexose sugar. Disaccharides are when two rings are connected together. This is a bond that is connecting them together. Our example is sucrose. You want to make sure you look in your lab for other examples. And then our last type of carbohydrate is called polysaccharides. They are very large, made of hundreds or thousands of glucose units connected together in a very long chain. This third type, which is called a polysaccharide, is often called a complex carb because it is pretty large. Whereas these two, monosaccharides and disaccharides, are both called simple sugars because they're much smaller. Let's move on to question number 8. Question number 8, we talk about different types of lipids. The first type of lipid to talk about is something called a phospholipid. The phospholipid is a component of all cell membranes. So its major function is to serve as a component in cell membranes. It is the major component of cell membranes. We want to know, what do we need to make a phospholipid? What is it made of? It is made of one phosphate head plus two fatty acid tails. And we will talk in more detail about what fatty acid tails are on the next page. At this point, I want to draw the symbol of a phospholipid so that you know what it looks like if you see it in your lab book or your online textbook. It looks like a circle with two squiggly lines connected to it. The circle represents the phosphate head, which is what we call hydrophilic. That means it loves water, and it is also polar. So we want to know those characteristics about the phosphate head. The two squiggly lines that are connected to the phosphate head are called the fatty acid tails. And like I said, we will go over the structure of them in a little bit more detail on the next page. Fatty acid tails are hydrophobic. They don't like water, and they are also non-polar. So please keep those terms in mind. So you can see that a phospholipid looks nothing like a glucose, which looks like a hexagon shape, or a fructose, which looks like a pentagon shape. So we want to get used to seeing the symbols of these different chemicals because that will be useful in chapters to come. The second major type of lipid is called a steroid. So the steroid is the second category or type of lipid. We will not continue to talk about this in a lot of detail, but I do want to mention one type of steroid is called cholesterol. It serves many functions, but one that I want to mention is that we do find it in cell membranes. There are two different types of modified cholesterol that I would like to mention. So the body will oftentimes take a chemical and modify it based on the demands of the body. One example of a modified cholesterol is estrogen. And the major function of estrogen is, of course, reproduction, as well as secondary female characteristics, physical characteristics, and it helps to build bone mass. During aging, estrogen decreases. So estrogen levels go down when someone ages. This may have an effect on bone mass being maintained. So as estrogen levels decrease, bone mass may also decrease, which could make an individual more susceptible to diseases like osteoporosis. The other type of modified cholesterol to mention is called testosterone, which again, its major function is reproduction. We want to mention that testosterone is also needed to build muscle mass. Build muscle mass. Building muscle mass can be an attractive quality for testosterone, and therefore supplements of testosterone may be taken or abused. You may have heard something like this in the news or read it online. Abuse of testosterone is usually called anabolic steroid abuse. Let's just mention anabolic steroid abuse, which is basically taking steroids which are not under a physician's care. This is more or less a push of testosterone into the body. Let's talk about the effects of that. First of all, anabolic steroids do increase or build muscle mass. Unfortunately, it may increase cardiac, which is in your heart, muscle mass, and that is abnormal. An increase in muscle mass is called hypertrophy. So basically, if an individual takes anabolic steroids or abuses anabolic steroids, they are at risk for their heart to hypertrophy, which throws off the structure of the heart and can get in the way of blood circulation and affect blood pressure and all sorts of other things. On the other hand, anabolic steroid abuse may also decrease the use of testes. The testes are an organ seen in males that produce sperm. And anytime you are decreasing the use of any type of organ, okay, you are at risk to shrink that organ, which is called atrophy. This is also going to decrease the production of sperm, which could lead to decreased fertility. Decreased fertility. Okay, we have to move on to the next page. So let's move on to question number 9. I want to go ahead and mention question number 9 is going to be in your lab book. So this you will have to know when you are working in the lab. This type of lipid is called a triglyceride. Triglyceride. So you can check the biochemistry lab in your lab manual. You will need to know what the triglyceride is made of. The triglyceride is going to be made of 3 fatty acids plus a chemical which is called glycerol. So if you want to make a triglyceride, you would need 3 fatty acids plus a glycerol subunit. You would connect them all together and you would produce the triglyceride. Triglycerides are basically useful as energy. They serve as a very concentrated source of energy. Fats are a better source of energy compared to carbohydrates because fats can store more energy per gram. If you had a gram of fat and a gram of carb, the fat is going to have almost twice the amount of energy stored in it. So therefore, fat really concentrates energy. However, your cells would prefer to metabolize the carb rather than the fat. Many cells need energy quickly. Metabolizing the carbohydrate happens much faster than metabolizing the fat for energy. This is the concept behind a low carb diet. Please always refer to your physician for anything like that. Let's draw the symbol of the triglyceride so that you can see how it does not look like a phospholipid or the carbohydrates in the previous examples. It looks like a very long letter E. The backbone of the E is called the glycerol. The glycerol unit or the glycerol backbone, and then the 3 long prongs extending, making the very long E, are called the 3 fatty acid tails. Let's take a portion of the fatty acid tail. So let's just draw one fatty acid tail in a little bit more detail down here. We're going to take a segment of that fatty acid tail that I highlighted with the green highlighter, and we're going to draw that portion of the fatty acid tail down here. Fatty acid tails look like carbon chains. Remember, we talked about that on the first page. A long carbon chain where it's basically all of the carbons in the chain are surrounded by hydrogens. So the fatty acids are primarily composed of carbon and hydrogen, and often get the term hydrocarbon applied to them because they are simply made of hydrogen and carbon. There are 2 types of fatty acids that you have heard about before. They're called saturated fatty acids and unsaturated fatty acids. The saturated fatty acids contain all single bonds and tend to be solid at room temperature. Examples are butter and coconut oil. Anything that's solid at room temperature, which is approximately 70 to 72 degrees. Let's draw a portion of a saturated fatty acid tail. So the saturated fatty acid tail is going to be made, again, just like a fatty acid, a chain of carbons that is going to have all single bonds between the carbon atoms. So we'll highlight those single bonds, all single bonds between carbon atoms within the carbon chain. So the two major things that you want to know about saturated fats have all single bonds and they're solid at room temperature. I like to remember the 3 S's. The last type of fat that we want to talk about is called an unsaturated fat. Let's talk about how it's a little bit different than a saturated fat. Again, we're going to draw a fatty acid tail. So we're just going to take a little portion. We're going to draw the chain, except now we're going to draw a double bond. So do you see how these little 2 lines here represent a double bond? If there is a presence of at least one double bond in the carbon chain within the fatty acid, then it's called an unsaturated fat. So unsaturated fats have at least one double bond, and they tend to also be liquid at room temperature. So our examples here would be vegetable oil or olive oil. What you want to recognize is that the presence of a double bond makes the fatty acid not consistent in shape. So if you look up here at the saturated fat, the fatty acid is pretty much straight. It has a very consistent shape. And anytime molecules have a very consistent shape, they tend to be able to stack on top of each other, which essentially ends up in them resulting in being solid at room temperature. When you have unsaturated fats, there may be presence of double bonds at different areas within the fatty acid chain. Therefore, you may not be able to sit one unsaturated fatty acid on top of another one due to the inconsistency that is produced by the presence of a double bond. And anytime molecules have a very inconsistent shape and we cannot stack them, they tend to be a little bit more disorganized in the volume of space that we're given, which gives them the characteristic of being liquid at room temperature. We are almost finished. We need to move on to the last group of organic biomolecules that we're going to talk about is going to be called proteins. We have a lot of different types of proteins. Let's give some examples of proteins that you've heard about before. Collagen, hemoglobin. It's okay if you haven't heard about them. Antibodies. There are so many different types of proteins out there. We have 23,000 different types of proteins in our bodies just to start with, and scientists speculate there's more. All of these proteins have different and vast functions, so it's hard to say what the function of a protein is because each individual protein has its own specific function. The monomers that are needed to synthesize or build proteins are called amino acids. Recall, those are the monomer subunits that are needed to build a protein polymer macromolecule. Let's draw the basic structure of the amino acid. There is a central carbon. I'm going to list that one over here. There is my central carbon. By the way, there are 20 different amino acids out there for us. And this is our central carbon. It has a very basic carbon skeleton. We're going to attach a hydrogen to our central carbon. That's step 2. We're building an amino acid, the basic structure of amino acid. The third step is to add a carboxyl to the second position on the central carbon. So I'm going to add a carboxyl. We've talked about that already back in question 3. That's my carboxyl group. If you look back at our functional groups, remember that this is also called a carboxylic acid. Step 4 is on the other side of the central carbon. We're going to add an amino group. Add amino group to central carbon. And at the last position of our central carbon, we're going to add something called an R group. R stands for various. I'm going to use the green highlighter here to circle that. So we're going to attach the R group to the fourth position of the central carbon. Okay, so the reason I highlighted the R group, okay, is because we can add different things at this position and make 20 different amino acids. So all 20 different amino acids have the central carbon, hydrogen, carboxyl, and amino groups. The only thing that is different between the 20 amino acids would be the R group. Let's give an example of that on the next page. There are 2 amino acids. Our first amino acid is going to be called glycine. So we're going to give 2 examples of amino acids, although on the last page, remember we said that there are 20 amino acids. And I am going to draw the structure of those amino acids. Our first is an amino acid, which is called glycine. So there I wrote it in big letters, glycine. The first thing I'm going to draw is my central carbon. I will attach a hydrogen. I'll attach a carboxyl group, just like I did on the previous page. I'll attach my amino group. And then at the fourth position, I'm going to attach my R group. Well, my R group in this case is going to be hydrogen, and that's it. It's a very simple amino acid. Let's draw another amino acid, which is called alanine. We're almost done here. I'm going to, again, draw my central carbon. So that's the same as in both examples. I'm going to draw my carboxyl group. So that's the same. I'm going to draw my amino group. So you can see that's the same as the other examples. You compare them side by side. They both have central carbons. They both have that hydrogen attached. They both have the carboxylic acid attached. They both have the amino groups attached. And the only thing that's different is going to be what I plug in at the fourth position to the central carbon. Well, now I made a little bit more complicated type of amino acid. Instead of plugging in just a hydrogen, I plugged in something called a methyl group. Remember, when we talked about the functional groups, we talked about methyl groups. So I plugged in a methyl at the fourth position, and that means I made a different type of amino acid called alanine. If I take the methyl group out and I plug in the hydrogen, then I will make an amino acid called glycine. A lot of amino acids end in ene. For example, phenylalanine, isoleucine, alanine, glycine. A lot of the names of amino acids end in that ene, but not all names of amino acids end in that. You need to look in your lab book, in your biochemistry lab, to check out other amino acids that are mentioned. Remember, this is under the category of proteins because amino acids are needed to make up proteins. And our last question, number 10, we are actually going to do in class together. So make sure you have completed everything before that, and we will do number 10, and then you will work in your lab groups to complete the rest of this handout, as well as the biochemistry lab. All of the material that we talked about is found in chapter 3 in your online textbook if you need a resource.